Okay, here's the Cliff's Note version for the people who don't want to watch the 20-minute video. This is the various methods by which monoclonal antibodies can be produced. First is the hybrid mouse hybridoma model. What you do is you inject with the target or the antigen into a mouse, then you harvest the splenocyte. So the spleen is where the B cells are. You screen them, you find the one that you want, then you hybridize it with a multiple myeloma cell to immortalize it, and then you get a mouse um, monoclonal antibody. Then you can chimerize it by putting the human portion in the back and make this a mostly human constant portion with a mouse variable portion. Or you can do what's called a CDR graft and you humanize it so it's mostly human and has some the mouse parts that recognize the antigen. And then most of because these cause an immune response in people with repeated usage, because we recognize this as foreign proteins, we're moving more towards human antibodies, and there's three ways of doing that. One is with a phage display, and this is like a little envelope that has the genetic material in it, but these phages display the antibody on the surface. Then you can have them you can screen them by seeing which one of them will stick to your target. And then the ones that stick to your target, you construct the human IgG and you express it in a transgenic system, transfection system. This is a transgenic mouse that basically has a humanized immune system. So it's like this mouse, this is a mouse that has a mouse immune system. This is a mouse with a human immune system, and then you inject them, you process them just like you do the mouse. But when you do this, you end up with a humanized antibody instead of a mouse antibody, a human antibody. Um, this one, the problem is that the antigen processing is mouse antigen processing and not human antigen processing. So these antibodies sometimes are not quite as good. Then a single B cell, what you do is you extract blood from a person that has been exposed to the target that you want. Um, and then you screen them. You sort the cells with labeled antigens using a flow cytometry machine, and then you figure out which of these cells from that donor produces the antibody that you want. And then you extract the DNA out of them and you place it into a plasmid by using PCR and other molecular biology techniques. And then you transfect them into a production cell line. And that's how the various ways you make a human antibody. If you want the long version, there is another video with the long version.